Hello everyone, here we have our star-studded cast. So let's start with our first actor, who is frizzled receptor. It plays an important role in Wnt pathway, as the pathway starts when the ligand basically attaches to it. Next we have LRP6, which stands for low-density lipoprotein receptor-related protein 6. It comes from a family of co-receptor and plays the role of a supporting actor, or aka a co-receptor of frizzled protein. Next, we have Wnt protein, disheveled protein, and beta-catenin protein. Wnt protein comes from a family of glycoproteins whose main role was to activate different intracellular signaling transduction pathways. As for the disheveled protein, it is a type of phosphoprotein which is involved in both Wnt pathways. Beta-catenin 1, or catenin beta 1, is a multifunctional protein that plays a role in signaling transduction and intracellular adhesion. Here comes the destruction complex. How can we forget about it? As the name suggests, the destruction complex plays role in destructing beta-catenin proteins through phosphorylation and ubiquination. This gang consists of APC, axin, GSK3, CK1, and beta-TRCP. Next, we have another unsung hero who likes to stay low-key. This barrel-shaped organelle is none other than proteasome, who plays the role of an assassin in this play. It gets rid of all the bad guys, including damaged or unintended protein.
And here comes our main lead, aka the big boss of the cell, which is also commonly known as DNA. It is the mastermind behind all the activities of a cell. Now moving on to the next introduction, we have TCF here, whose full name is T-cell factor enhancer factor. But I think it is only called by the full name when it gets scolded for messing up the transcription. Last but not the least, we have Groucho here, who keeps our naughty TCF from making mischief. Acting as its corepressor, it keeps tight control over TCF activities.
In the inactivated form, the destruction complex controls the beta-catenin levels in the cell. It associates with the beta-catenin and leads to the phosphorylation by GSK3, and that signals ubiquitination of beta-catenin by beta-TRCP. This leads to proteasomal degradation of beta-catenin, and the cellular levels of beta-catenin become low. In activated form, Wnt comes and binds to frizzled. Activated frizzled leads to the phosphorylation of LRP. It causes the translocation of the complex near the frizzled and LRP in the membrane. DVL binds to LRP and gets activated. Activated DVL inhibits the destruction complex, and so beta-catenin is not phosphorylated. This does not allow the beta-TRCP to inculcate into the complex, and thus no ubiquitination. There are high levels of beta-catenin in the cell. The TCF is present in the nucleus. Now let's discuss Wnt pathway in the cellular processes. Wnt pathway is involved in cellular polarity. Cellular polarity refers to the intrinsic asymmetry observed in cells, either in their shape, structure, or organization of cellular components. It is also involved in cell fate determination. It helps the cell to develop into a final cell type. Furthermore, this pathway helps in movement of cell. It also helps in organogenesis. Organogenesis is the phase of embryonic development that starts at the end of gastrulation and continues until birth. During organogenesis, the three germ layers formed from gastrulation, the ectoderm, endoderm, and mesoderm, form the internal organs of the organism. It play role in stem cells renewal. Involvement of Wnt signaling pathway in cellular processes. First of all, Wnt pathway is involved in different cellular processes, especially during embryonic development, which was first found operating in the segment polarity and body axis formation of Drosophila, where it helps to establish anterior and posterior polarities, anteroposterior and dorsoventral axes. So, Wnt signaling functions can be divided into axis patterning, cell fate specification, cell proliferation, and cell migration. So, the first cellular process in which Wnt pathway is involved, as axis patterning Wnt signaling activity, in axis development can be seen in mammals, fish and frogs. In mammals, the primitive streak and other surrounding tissues produce the morphogenic compounds like Wnt's and retinoic acid to establish the posterior region during late gastrula forming concentration gradients. Highest concentration areas establish posterior region while lowest concentration areas designate anterior region. In fish and frogs, beta-catenin produced by canonical Wnt signaling causes formation of organizing centers. The second cellular process is cell fate specification or differentiation. Wnt signaling induces differentiation of pluripotent stem cells into mesoderm and endoderm progenitor cells, which further differentiate into cell types such as endothelial, cardiac and vascular smooth muscle lineages. Wnt1 provokes neural differentiation, which is a key factor in self-renewal of neural stem cells that allows for regeneration of nervous system cells. It is further evidence of a role in promoting neural stem cell proliferation. Wnt3 leads to mesoderm-committed cell with hematopoietic potential. Wnt signaling also antagonizes heart formation, and its inhibition is a critical inducer of heart tissue during development. Small molecule Wnt inhibitors are routinely used to produce cardiomyocytes from pluripotent stem cells. The third one is cell proliferation. Proliferation and growth of embryonic stem cells is facilitated through canonical Wnt signaling, which increases nuclear and cytoplasmic beta-catenin. Increased beta-catenin can initiate transcriptional activation of proteins such as cyclin D1 and CMYC, which control the G1 to S phase transition in the cell cycle, as entry into the S phase causes DNA replication and eventually mitosis that are responsible for cell proliferation. The fourth one is cell migration. Cell migration during embryonic development allows for the establishment of body axes, tissue formation, limb induction and several other processes which are mediated by Wnt signaling, particularly convergent extension. Wnt signaling also induces cell migration in later stages of development through the control of the migration behavior of neuroblasts, neural crest cells, myocytes and tracheal cells. Wnt signaling is also involved in another key migration process known as the epithelial mesenchymal transition, EMT, which allows epithelial cells to transform into mesenchymal cells so that they are no longer held in place at the laminin. So, the last but rather an important cellular process mediated by Wnt signaling is insulin sensitivity. The process of upregulation of glucose transporters in the cell membrane in order to increase glucose uptake from the bloodstream is partially mediated by activation of Wnt beta-catenin signaling that increases a cell's insulin sensitivity. Especially, Wnt10B is a Wnt protein that increases this sensitivity in skeletal muscle cells. Normally in Wnt pathway, there is a CYS-rich domain due to which ligand bind to the 7-transmembrane domain. But due to absence of CYS-rich domain, Wnt ligand would not bind to the receptor and the mutation start. Axin served as scaffold for the formation of destruction complex which would degrade beta-catenin. Then comes APC, which is also known as Adenomatous polyposis coli, which interacts with beta-catenin and axin at a time and hold beta-catenin in a destruction complex. The other components CK1 alpha and GSK3 beta phosphorylates the beta-catenin. Then beta-catenin is target by BTRCP and transfer ubiquitin chain to beta-catenin. Beta-catenin will be then ubiquitinated via proteasomal degradation pathway. Meanwhile, in nucleus Wnt target genes transcriptional factors, which is TCF, in absence of beta-catenin would bound to Groucho, which is transcriptional repressor, and thereby no target gene could be expressed. And now we will discuss about the diseases involving the Wnt pathway. Wnt pathway is one that is an evolutionarily conserved and plays a tremendous role in developmental, proliferative, and immunological functions. There are a number of pathologies that result from mutations in the Wnt pathway disruption. These include tetra amelia syndrome, Fuhrmann syndrome, type 2 diabetes. Various mutations associated with the Wnt signaling cascade also result in odonto-angiodermal hypoplasia, hyperparathyroid tumors, and early coronary disease and osteoporosis. Wnt pathway disruption is known to have a special role in oncogenesis. Mutations in the Wnt pathway cause adenomatous polyposis and cancer, which are due to the mutation in the APC gene, colon cancer, Wilms tumor, sebaceous skin tumor, etc. We will only discuss some of these pathologies here. The first one is the tetra amelia syndrome. It is characterized by a total absence of all four of the limbs. It is caused by a mutation in the WNT3 gene. A homozygous WNT3 nonsense mutation in the chromosome 17 leads to anomalies, since the said gene plays great role in the embryonic development. WNT3 is required at the earliest stages of limb development and craniofacial and urogenital development. A rare case of consanguineous family with 4 affected fetuses displaying recessive tetra amelia and craniofacial defects were studied. Wilms tumor or nephroblastoma is a form of kidney cancer primarily in children aged 10 and below. WTX is a Wilms tumor-associated tumor suppressor protein, and a single mutation in its gene is enough to cause carcinogenesis. WTX lacks the conserved functional protein domains. Absence of WTX leads to the rapid degradation of NRF2, a transcription factor and key control point in antioxidant response. Among its many functions, WTX also regulates the stability of beta-catenin, and modulates the activity of WT1, Wilms tumor 1, transcription factor in the nucleus. Type 2 diabetes mellitus is characterized by insulin resistance. In case of type 2 diabetes mellitus, studies revealed that the gene-gene interaction between downstream gene glucagon and polymorphic LRP5 gene. Positive feedback between glucagon-like peptide, GLP-1, and Wnt signaling pathway leads to increased proliferation in the pancreas beta cells and suppression of apoptosis.